Good morning, good afternoon, good evening, everyone. Thank you all for being here today. My name is Sriram Maravamudan. I'm a key account manager and proteomics product specialist at Cell Signaling Technology. Welcome to today's webinar on a highly sensitive post-translationally modified peptide enrichment for LC-MS analysis, cleaner immunoprecipitation, reduced sample requirement, and simplified sample preparation for ubiquitination and SUMOylation. Firstly, I'd like to introduce our speakers for today, Dr. Alissa Nelson and Dr. Jeroen Demmers. Dr. Alissa Nelson is a senior proteomics scientist at Cell Signaling Technology. Alissa earned her PhD in biochemistry from the University of Notre Dame in South Bend, Indiana, USA in 2016, studying with Dr. Amanda Hummon. She worked on new methods of phosphoproteome sample preparation and applications to colon cancer biology. From 2016 to 2017, she was a postdoctoral scholar in the Proteomics core facility at Northwestern University in Chicago, Illinois, supervised by Dr. Yang Agu and Dr. Neil Kelleher. Since 2017, Alissa has worked at Cell Signaling Technology, providing proteomic services and developing new products specialized in the analysis of post-translational modifications. Our second speaker for today, Dr. Jeroen Demmers, is the director of the Erasmus Medical Center Proteomics Core Facility in the Netherlands. Jeroen studied chemistry at Utrecht University in the Netherlands. He earned his PhD degree from the same university in 2002, working on the development of tools to study interactions of transmembrane peptides and proteins with phospholipid bilayers using hydrogen deuterium exchange and electrospray ionization mass spectrometry in the groups of Albert Heck and Antoinette Killian. During his postdoctoral studies in the group of Brian Scheidt at the Rockefeller University in New York, he worked on a project involving the proteomic identification of plasmid DNA binding proteins in yeast. In 2005, Jeroen moved to Erasmus Medical Center with a Euro Fellowship to initiate a mass spectrometry and proteomics facility and to set up a line of research in the field of protein mass spectrometry. Currently, he is an assistant professor of proteomics, and his research focuses on the molecular mechanisms of the ubiquitin proteasome system, for which he develops quantitative proteomics technologies that focus on the analysis of protein post-translational modifications. Jeroen is the director of the Erasmus Medical Center Proteomics Core Facility and collaborates with many research groups within the institute and abroad. Today, we will introduce an improved immunoaffinity enrichment method for KGG peptides, which is our PTMScan HS workflow that significantly improves sensitivity, specificity, and robustness of ubiquitinated peptide identification and quantification. You will also have the opportunity to ask questions by typing them into the GoToWebinar questions area in your control panel. Without further ado, I'll turn it over to our proteomics expert, Dr. Alissa Nelson.
All right. Hello, everyone. Thank you so much, Sriram, for that introduction. Today, I'd like to share an overview of the PTMScan method, how it applies to ubiquitin and SUMO proteomics, and then talk about our efforts to redesign PTMScan from the ground up to make it more sensitive and easier to use. So Cell Signaling Technology has been developing and manufacturing antibodies in Massachusetts in the United States since 1999. We're recognized by our peers for our environmental values, our community involvement, and charitable contributions. We're a company made up of scientists who are motivated to provide our customers with the best possible quality products that will allow you to do excellent science yourself. So about 15 years ago, Cell Signaling Technology pioneered the PTMScan method as a way to enrich for post-translationally modified peptides. Cells or tissues can be lysed in 9 molar urea, digested by appropriate enzymes. Those peptides are purified prior to performing an immunoprecipitation specific for those PTMs. We analyze the peptides by LC-MS, and we can perform peptide identification and quantification. After all these years, we've released approximately 35 different antibodies in this product line, and some of those include phosphotyrosine, acetylysine, monomethylarginine. Dimethylarginine comes in both asymmetric and symmetric flavors, lysine methylation, as well as phosphoserine and threonine in all sorts of different kinase motifs. And the focus of the talk today will be on our products for ubiquitin and SUMO remnants. So while I'll spend the bulk of the time today talking about reagents for you to do this type of analysis in your own labs, I also just want to point out that CST also offers a services department. So you can send your biological samples of interest to us, and we'll send you back a report with all the results. So the original principle for studying ubiquitin site modifications was pioneered as a collaboration between CST scientists, including Dr. Ailan Go, and academic research groups. So as many of you know, ubiquitination is an important post-translational modification that signals proteins to be degraded. Abnormal ubiquitination is involved in cancer, neurodegeneration, metabolic syndrome, and other diseases. If this blue squiggle represents a substrate protein, the lysine that's modified branches off with this GG followed by an arginine, and this sequence can be cleaved by trypsin that yields this branched GG remnant. And this is what we can enrich for using a PTM specific antibody. The principle for SUMO site profiling is very similar. So again, this was work that was carried out by both CST scientists and academic researchers. So the sequence for SUMOylation is a little different than ubiquitination. There's a threonine at this site rather than an arginine. So it's not cleaved by trypsin, but it can be cleaved by wild-type alpha-lytic protease, or WALP, and again can yield this GG branched remnant. SUMO, of course, is important in multiple biological processes that include, nuclear transport, DNA replication and repair, mitosis, and various other kinds of signal transduction. So while the PTMScan technique has been used to publish a multitude of valuable papers over the years, we in the services group and our customers, we've noticed a few pain points with this workflow. So not so long ago, the development team at CST set out to completely redesign the PTMScan workflow to be high specificity, high sensitivity, and high simplicity. So some of the goals that we began with were, number one, to increase the sensitivity of this workflow and reduce the sample input requirement. So currently for PTMScan kits, it takes anywhere from 5 to 15 milligrams of protein input to obtain a meaningful result. And we sought to cut this down to just 1 milligram. We've also set out to remove the requirement to have a lyophilizer. And part of that is just scaling down the amount of material that needs to be dried. We wanted to reduce the amount of time that's required for sample preparation. You'll notice in this image that it takes 4 days to get to purified, clean peptides that are ready to immunoprecipitate. So there's a lot of room to save time with this. We also wanted to move away from using urea, as this can cause carbamylation side reactions, and that's really undesirable. One of the major things that we wanted to fix is that the antibody would elute off of the agarose beads that come in the kit. And this can end up interfering with the LCMS analysis. And then finally, we also sought to increase the specificity of the enrichment and reduce the number of unmodified peptides that come through in the final sample. So the first major change that we made to this workflow to accomplish those goals was we changed the urea digestion to instead use S-traps, which are provided by Proteify. So the first part of this is that the lysis buffer was changed from urea to 5% SDS, and this helps to improve protein recovery. The protein lysates are loaded onto S traps, which are just a quartz spin column. They're precipitated there. They're washed with several rounds of methanol in order to remove all the contaminants, and then the digestion can be performed directly on the cartridge. The washing steps only take about 15 minutes, and then the elution steps the next day also only take about 15 minutes. So this saves a lot of time compared to performing a C18 set pack purification. The final sample can be dried in a speed back overnight, so you no longer need a lyophilizer. And altogether, this means the sample preparation time has been reduced from 4 days down to just 2 days. We wanted to carefully evaluate the differences between urea and S traps before making a permanent change. So we noticed when we performed the BCA assay on our lysates that S traps, the S trap lysis buffer, the 5% SDS, routinely yielded more soluble protein than urea buffer did. So across multiple different cell and tissue types, we see anywhere from a 10% improvement to about a 35% improvement. We've also noticed that SDS can better solubilize certain types of modified proteins. While this doesn't happen in every case, we did notice that this SDS lysate compared to the urea lysate had more ubiquitin as well as more SUMO 2 and 3 by Western blot. When we perform total proteome analysis, we see that S traps enhance the digestion efficiency and actually maximize the number of fully tryptic peptides that we can identify, which is shown in this dark blue bar here. And then finally on the right, all of these things combine to mean that we're able to identify a very high number of KGG peptides using S traps, and it at least matches or improves the performance compared to the old preparation method. The real core of this new protocol is the immunoenrichment, and this work was primarily carried out by my colleague, Dr. Yiying Zhu. So her first major innovation here was to find a way to permanently couple the antibody to the beads. This eliminates the potential to clog your LCMS column when the antibody would elute from the beads. Now the antibody doesn't elute. She also tested something like a dozen different types of beads in order to find the perfect magnetic bead that would reduce non-specific binding, and mag beads, of course, are very popular because they're so easy to handle compared to agarose. Finally, she carefully optimized binding and wash buffers in order to minimize the background peptides, and this really increased the enrichment specificity. So I'd like to emphasize a little more the importance of keeping the antibody coupled to the beads. So in the classic PTMScan kits, we use an agarose bead, and the antibody is, of course, non-covalently coupled. And when we elute with acid to remove the peptides, the antibody also elutes off of these beads. So you can see by Western blot, this TFA eluent contains quite a lot of antibody, and even after we perform a stage tip, we've reduced the amount of antibody, but some of it can still remain. So there ends up being an awful lot of work involved just to make sure that this intact antibody doesn't cause a problem in your LC system. So the new HS antibody kit has the antibody permanently coupled to the magnetic bead, so it does not elute when we add acid. And you can see from this graph we've quantified that using agarose beads, essentially all of the antibody would elute, but with the new magnetic beads, the amount of antibody coming off is negligible. So we evaluated the performance of the antibody using peptides that were all prepared the same way. These are mouse liver peptides prepared by the classic urea sample preparation. So you can see that in this blue bar, 1 milligram of peptide input enriched with the classic KGG antibody yielded not quite 3,000 KGG peptides, but the new antibody was able to recover double the number of KGG peptides. Also, you can see that even with 5 milligrams of input, the classic antibody was not able to match that performance with just 1 milligram put in with the new antibody. We also see a big improvement in specificity as well. These blue triangles show that the classic enrichment essentially is somewhere close to 15% specificity. So when I say specificity, what I mean is the number of ubiquitinated peptides divided by the total peptides that were identified in that enrichment sample. So going to the new antibody, we're able to increase that number to 50%. When we combine the S-trap digestion procedure with the PTMScan HS antibody, we're able to see really stark results, improving both the sensitivity and the specificity of the assay. So on the left, we have MKN45 human gastric cells. So we're seeing double the number of peptides being enriched by this combined new digestion and new enrichment procedure. And the specificity has gone up by double as well. When we evaluate mouse tissue, mouse brain tissue with 1 milligram of input, again, we're seeing better than double improvement in specificity, in sensitivity, sorry, and the specificity is nearly quadrupled. We were able to validate that this workflow works for SUMO as well. So on the left, you're seeing HCT 116 cells that are prepared by all the same peptide preparation method, by the Urea method. And we're comparing the classic antibody versus the new antibody. So that new antibody in a SUMO context does improve the sensitivity, the number of SUMO peptides that are identified, and it also gives a great improvement in specificity as well. When we performed an experiment to compare the digestion methods using the new KGG antibody to perform all of the enrichments, we saw a really striking difference between the performance of the Urea and the S-traps for the case of SUMO. So not every SUMO experiment we've performed has had quite this striking of a difference, but you can see these Western blots confirming in the lysates just how much more modified proteins were extracted by the SDS lysate. And this translates to a huge 20-fold improvement in the sensitivity of the pull down. And even at 5 milligrams of input, that Urea lysate is not able to identify a meaningful number of SUMOylated sites. So just to sum up, the classic PTMScan kits did require up to 15 milligrams of protein input, but now with the HS kits, 1 milligram of protein is plenty to perform interesting biology experiments. With the classic kit, there was a large-scale in-solution digestion, and the workflow took 4 days or more. With the HS kits now, S-traps allow the peptides to be prepared in 2 days, and it maximizes the recovery. Agarose beads were messy to handle, and they would bind unmodified peptides non-specifically, but the new magnetic beads are much easier to handle, and they reduce the background binding. Antibodies used to elute from the beads right along with the PTM peptides, and that tended to cause problems for the LC systems, but now that they're covalently linked, the systems are much cleaner. And I just want to emphasize that, that the underlying antibody hasn't changed, that this is still the same proven CST validated antibody that's at the core of this method, and you're still getting that same underlying antibody when you switch to the new kit. if anyone's interested in making a purchase, there is, this kit is released, it's on our website, catalog number 59322. That kit comes with the magnetic beads, with the antibodies, of course, and the binding and wash buffers that you need to perform the enrichment. We're also working on launching a sample preparation companion kit with the S-traps, and that's due to come out at the beginning of 2021. So in the short term, customers are free to order S-traps directly from protify.com. And certainly if you're very familiar with the urea sample prep method and you like that method, you can continue to use that in the short term. So with that, I'd like to thank all of my teammates who helped in the development of this new kit. It took quite a lot of people to get it all validated and released. So with that, I will turn things over to Jeroen.
Thanks, Alissa. Sorry about this. And thanks, Shriram, for the kind introduction in the beginning. And thanks for having me today. So I'm going to tell you about some of the work that we do on the analysis of protein ubiquitination using the PTM enrichment kits provided by CST, of course. And I will also give you some of the results of our initial experiments with these new HS beads. And I must say that the first results really look spectacular. I have to admit that we haven't been able to do all the experiments and analysis that we had in mind because of the, well, at least partial closure of the lab and the absence of some co-workers. I'll give you some preliminary results anyway, but please be aware that much of it is still a work in progress. Just as a brief introduction, my lab is the research core facility for proteomics at Erasmus Medical Center, Rotterdam. We collaborate with many different departments in our institute and abroad and on really different topics. Besides that, we run our own projects, many of them on the analysis of protein post-translational modifications. Ubiquitin was already nicely introduced by Alissa, but briefly, the 20S proteasome is a highly conserved protein complex and part of a major mechanism by which cells regulate the concentration of particular proteins and degrade unneeded, damaged, and misfolded proteins. Proteins are tagged for degradation by ubiquitin and then sent to the proteasome for degradation. Abnormal UPS functioning has been implicated in the pathogenesis of many human diseases, including cancers and neurodegenerative disorders, such as Alzheimer's and Parkinson's diseases. The idea here is that ubiquitinated proteins targeted for degradation cannot be degraded anymore by the impaired proteasome activity and cause neurotoxicity this way. There is a rising interest in the UPS as a pharmacological target to prevent and treat neurodegeneration, as counteracting the inhibition of the UPS may have beneficial effects. The proteasome is also a target in cancer therapy, for instance, for multiple myeloma. And the idea here is that cancer cells go into apoptosis when the proteasome is inhibited. So ubiquitin is a post-translational modification that is conjugated to the epsilon amino group of lysines. And a key feature of this PTM is its ability to form a polymer change in which individual moieties are linked via one of 7 lysine residues or the N-terminal methionine. Only few of those distinct linkage types have been studied extensively, mainly K48 and K63, but not much is known about the other linkage types. And the idea is that these may be involved in all kinds of signaling events in the cell. So we are interested in dissecting the roles of individual subunits of the proteasome to understand the molecular mechanisms of the UPS. And for this, we manipulate the proteasome by depleting individual subunits, in this case in Drosophila S2 cells, but we are now also extending this work to human cell lines. And we set off by inducing complete inactivation of proteasomal activity. And for this, we have selected 3 key subunits simultaneously to make sure that all proteasome activity is abolished. We compare this situation then to inhibition of the proteasome by small molecule drugs, which target the proteolytic activity of the proteasome. We use a SILAC mass spectrometry-based approach, including helic fractionation at the peptide level to identify and quantify the proteome. And in addition, we focus on the ubiquitinome, the complement of all ubiquitinated proteins in the cell, by specific enrichment of diglipeptides. And diglipeptides are produced when ubiquitinated proteins are digested with trypsin, leaving this specific diglycine remnant on the target lysine. The depletion of proteins in fly cells is relatively simple and works by just adding an amount of dsRNA to the culture. And after 2 days of dsRNA treatment, protein expression levels already are at their lowest, and there's basically no difference with the levels after 4 days of incubation. And in the lower plot here, you can see that this results in the accumulation of ubiquitinated proteins, just like when the proteasome is inhibited by chemical drugs. So in these scatter plot of heavy to light ratios, SILAC heavy to light ratios, the 2-log ratios for both the forward and reverse, so experiments with including a label swap, for each of the identified proteins is plotted. And in control experiments over here, ratios are all centered around 1 to 1, meaning that the relative abundances remain unchanged. And then after inactivation of the proteasome with either inhibitors or RNAi mediated knockdowns, the protein abundances are increased either by synthesis or accumulation, indicated by the presence of proteins in the upper right quadrant over here. Up to 10% of all proteins increase more than 1.5 times in abundance. And strikingly, all proteasome subunits increase in abundance upon inactivation, both by chemical inhibitors as well as by subunit knockdowns. And this may be caused by some compensatory reaction induced by the loss of proteasomal activity. In the case of the triple knockdown, cells seem to die or go into apoptosis. So for all the downstream analysis, we have left these out of the calculations. There's quite some overlap between the upregulated proteins under the various conditions, as you can see here in this heat map. Fast responders include stress response proteins, and we know they are biosynthesized as a result of proteasome inhibition, so they are not accumulated but synthesized, and show increased abundances over prolonged periods of time. Further GO analysis shows that the population of proteins that increase in abundance represent categories such as the UPS, apoptosis, cell cycle regulation, and also stress response. If you now focus on diglipeptides representing ubiquitination sites, the effects are much more dramatic. So first, the distribution is much broader. And second, it's clear that the far majority, over 70%, of diglipeptides are really upregulated, as you would expect when ubiquitinated proteins are accumulated because they cannot be degraded anymore. The patterns are similar for the triple knockdown and for the inhibitor-treated cells. Now, this massive amount of detailed information on diglipeptides allows us to look at the dynamic changes of protein ubiquitination in relation to the global protein abundance changes based on the non-modified peptides. And on this slide, I show a few examples of proteins and their non-modified and diglipeptide ratios. And as you can see, in many cases, there is a correlation between the ratios, as you can see here. So a correlation between the gray non-modified peptides, the gray bars, and the diglide peptides, the orange bars. These show an increase in both ubiquitination and global abundance. However, many proteins show more complex patterns, where there is, for instance, an increase in ubiquitination accompanied by a decrease in total protein, or even opposite behavior of diglide peptides within the same protein, as you can see here or here. Now, two interesting examples are highlighted here, RPS3 and H2B. Although the non-modified peptides give consistent ratios, the diglide peptides show heterogeneous ratios. So ubiquitination seems to be up and down regulated in a site-specific manner here. If you now localize the distribution of these ubiquitinated lysines in these proteins, you can see that while most ubiquitin sites are upregulated in histone H2B, here in green, the one in red goes down. And now this happens to be a lysine residue, which is known to be monoubiquitinated and is involved in transcriptional activation. So we actually know that this ubiquitination site clearly has different function than being just a mark for protein degradation. In this ribosomal protein RPS3, a ribosomal building block that also has extra ribosomal functions, all the ubiquitination sites that are downregulated are specifically located in the C-terminal region, while the upregulated ubiquitination sites are all in the structured domains. Now the question obviously is, what does it mean? One possibility is that these diglide peptides arise from ubiquitination events of different linkage types and represent ubiquitin signaling of distinct functionalities within the same protein, and maybe no degradation marks. Another possibility is that we are in fact looking at different populations of proteins, for instance, at distinct subcellular locations. And a third possibility would be that this suggests a mechanism in which proteins are only partially degraded by the proteasome to obtain active protein domains. The total pool of ubiquitin itself is highly upregulated in all cases. And if you now zoom into the various ubiquitination sites of ubiquitin itself to look at the different linkage types of polyubiquitin, we do not observe clear differences between the 7 known sites. What we certainly do not observe is a stronger increase in K48, which you might expect because this has been described as the canonical degradation mark. And if proteasome activity fails, then this would be the linkage type that would be accumulated most. If you look at the spectral intensities for diglide peptides of ubiquitin itself, then these are orders of magnitude lower than those for the non-modified counterparts. So they are really lower in intensity. We then focused on the roles of the 3 different deubiquitinating enzymes, or DUBs, that are associated with the proteasome. DUBs clip off ubiquitin from the target protein, but the functional differences between them are not entirely clear. There are indications from previous studies that USP14 and UCHL3 may be editing DUBs that could clip off ubiquitin building blocks from the distal end of polyubiquitin, while RPN11 is the main DUB that cleaves off the complete polyubiquitin tail when the protein is still unfolded and drawn into the proteasome barrel. Now we applied our ubiquitinome screening method to study the functions and characteristics of these 3 different DUBs, which were knocked down individually, and we noticed some remarkable differences. Where the depletion of RPN11 causes a huge remodeling of the ubiquitinome, the depletion of UCHL3 and USP14 has apparently no effect. and neither had the double knockdown. So there are clear functional differences between these DUBs. And we are now trying to sort out whether it's possible to learn more about the targeting specificities of these DUBs. Then we made some improvements to the diglide peptide enrichment protocol by applying fractionation before doing the peptide IPs, performing multiple IPs per fraction, introducing more efficient wash steps, and including an effective decision tree in the Excalibur software. And this way we can now identify over 23,000 diglide peptides from just a single HeLa sample. And here you can also see that fractionation results in a high number of unique peptides per fraction. But I have to say that this work was all done using the old beads. And of course, we were very curious to see whether the new HS diglide beads would give us even better results with several updates and improvements in the protocol, as Alissa just told you about. And I think it's yeah, the new HS beads really have tremendous advantages. So usually when I talk to people about the possibilities of modified peptides enrichments, they are really interested, but also reluctant when they hear about the amount of starting material that is necessary to obtain decent results. So I think maybe the largest improvement would be that the new beads, for the new beads, you need much less input material than before. And this was actually the first thing that we tested in the lab, and the results were immediately obvious, I would say. So using an amount of only 250 micrograms of input material, and in this case, this is our benchmark sample of bortezomib-treated HeLa cells, we identified almost 3 times more diglide peptides using the HS beads as compared to the old beads. And next, we tried whether the novel wash buffer made a difference in the IP specificity. And indeed, it turned out that when using the new wash buffer, more diglide peptides were IP'd with less background of non-modified peptides. Those results are listed, are illustrated in this bar graph on the right. One of the areas of focus at the moment in our lab is to try to combine diglide peptide enrichments with TMT labeling. And for those of you not familiar with this type of labeling, I'll briefly go through the procedure. So TMT is a chemical moiety that is composed of a mass reporter group, a mass normalizer group, and a reactive group. And peptides can be labeled using TMT on their N-termini and their lysine side chains. TMT Pro, the new version of TMT, comes in 16 different flavors, and although their total masses are the same, they differ in the distribution of heavy stable isotopes. So all differentially labeled peptides end up at the same M/Z value in your mass spectrum, and only after fragmentation, the difference between each of the labels is visible in the reporter ion region in the mass spectrum. And from the reporter ion intensity, the original peptide abundance can be calculated. Now, one problem that occurs is that because of the gly-gly remnant that is conjugated to the lysine epsilon amino group by an isopeptide bond, an additional N-terminus is introduced in the modified peptide. And that would also be TMT labeled, leading to an increase in mass of the peptide. Now, this could be solved by performing the labeling reaction while diglypeptides are still bound to the beads. And in that case, the idea is that the antibody protects the N-terminus of the gly remnant from labeling. In comparison, if you label peptides in solution, then you would get this additional TMT label here. Now, this is indeed what you see in your in the mass spectra. So, for instance, this diglypeptide of ubiquitin itself, the K48 linkage peptide, you've seen this one before. It is by far the most intense peak in any digly IP. So, when TMT labeling is performed on bead, the peptide represented by the spectrum on the right will be the most abundant peptide. And in this case, this peptide is modified by only one TMT label on the N-terminus of the peptide, and not on the extra N-terminus on the gly remnant. And the reason why I'm showing you this is that the new magnetic beads are so much easier to handle. For instance, for TMT labeling, this has become much easier to do. And this is, I think, another big improvement of the new HS beads. Now, this slide shows the numbers of TMT modified diglypeptides. The number of peptide groups is somewhat overrated here because only partially TMT labeled peptides are counted as separate groups as well. But I think it's clear that the number of peptides in the TMT labeling experiment compared to the control shows that there's virtually no loss of diglypeptides when labeling them with TMT. And this is true for the new HS magnetic beads. And I will show you in a minute that when doing this with the old beads, the number of diglypeptides is much lower upon labeling. This is just to show you that what you get when TMT labeling takes place in solution. So in that case, the most prominent diglypeptide variant is the one that contains two TMT labels. And this is just to show what a TMT pro digly experiment looks like. Don't pay attention to the relative abundances. That's something that we still have to work on. This is just to show you all the 16 quantitation channels. And it's also obvious that the increase in total mass of the peptide upon labeling is quite substantial at an almost 1000 Dalton increase. We have used TMT labeling in a project where we look at targets of the deubiquitinating enzyme ubiquitin specific protease 7 or USP7. And this is a protein of immense biological importance as it is involved in several cellular processes, including host virus interactions, oncogenesis, tumor suppression, DNA damage, DNA repair processes, DNA dynamics, and so on. USP7 functions as a transcriptional co-repressor in the nucleus and binds to the metabolic enzyme GMPS. And this complex is then recruited to specific regulatory elements by gene-selective transcription factors. And then GMPS USP7 can promote gene repression, at least in part, by the removal of the active H2B ubiquitination mark. In addition to acting as a transcriptional co-repressor, GMPS USP7 can also modulate gene expression programs through deubiquitination of transcription factors, including the tumor suppressor P53, for instance. So several targets of this DOP are known, but we were interested in finding additional targets. And for this, we took a ubiquitinamics approach and along the line made a direct comparison between label-free quantitation and TMT labeling of diglypeptides. We took advantage of this project to compare these two types of quantitation. And in terms of numbers of peptides, in case of label-free quant, many more diglide peptides were identified as compared to TMT labeling. And I have to say that this is TMT labeling in combination with the old beads. And of course, what we really are planning to do now is to repeat these experiments using the new beads and use TMT labeling on bead. Anyway, these experiments showed that label-free quant still delivered more diglide peptides than in the case of TMT labeling. Now, these are the plots, the volcano plots of all data together, and you can see clearly a broader distribution for the label-free quantitation. But what's maybe more interesting now is when you zoom in into two proteins that I unfortunately have to anonymize here because it's all unpublished collaborative work. You can nicely see the reproducibility of for all the diglide peptides of these two target proteins. So in this case, the first 3 channels of 6, so this was a TMT 6-plex experiment. The first 3 channels are triplicates for the wild type, and the last 3 channels in red are triplicates for a USP7 knockout cell line. So basically, when you take away the ubiquitin protease, you get an increase in ubiquitination on these lysines. So that makes sense. And if you now directly compare the label-free and TMT quantitation for these diglide peptides of these 2 specific proteins, you can definitely see a similar trend, although the exact fold change numbers are quite different. And this discrepancy may be attributed to the so-called compression effect of TMT ratios. That's also something that we're working on right now. So in conclusion, using the PTMScan HS ubiquitin remnant beads, it is now possible to dig the deeper ubiquitinome with less starting material. And this is highly useful for studying heterogeneous ubiquitination in proteins, and of course for smaller amounts of starting material. Adaptation of the wash buffer leads to less background and more specificity. That's also something we have seen. And a big improvement, I think, is that the magnetic beads are much easier to handle, for instance, in case of TMT labeling. So I'd like to thank several people in the lab, and especially Karel Bestelhosti, who did most of the diglide peptide work over the past months. Thanks for your attention, and I'll give the control back to Jeroen.
Thank you very much. I would like to thank both our presenters today for their great presentations. We will now go through your questions. Please continue to type any questions you may have into the questions area in the control panel. Right, we can see our presenters. So I'm going to read out the first question. I believe it is for Dr. Nelson. Do the antibodies in kits work for plant samples, and do you have literature to share?
So the antibodies do work for plant samples, with the caveat that you should probably double check the sequences of your particular species of interest. So I believe that the that the RGG sequence is conserved in ubiquitin to Arabidopsis, for example, but you should probably double check your own species. So yes, the antibodies work for any species that has that sequence. And then as far as the S-trap digestion, I think there is some literature showing that plants can be digested using S-traps as well. Any digestion method that you have success with should be fine there.
All right, thank you very much. The next question would be, is the antibody covalently attached to the magnetic beads, or does it remain on the magnetic beads because of strong interactions between the antibody and the beads?
Well, the details of how the antibody is coupled to the beads are proprietary, but it is permanently attached. We've shown that data to indicate that the antibody is not leaching off of the beads during the elution step.
Great, thank you, Alissa. The next question would be for Dr. Demmers. For on-beads labeling, after labeling, you do digestion, and how do you quench completely?
Well, so in fact, the labeling is performed on bead, and we basically use the standard protocol for quenching that comes with the TMT kit. So there's actually no difference in doing in solution labeling.
Okay. I think there was another question related to that, which says, have you directly compared the KGG peptide ID in solution TMT labeling and on-beads TMT labeling? And is the on-beads better in this aspect?
Yes, I think we're still working on optimizing the on-bead TMT labeling, but as far as I can say on the results that we have obtained so far, it is better. Especially because the N-terminus of the diglycide remnant is not labeled, and that saves. So that makes peptides in general lighter. So the masses are lower, obviously, but also the software like Proteome Discoverer has difficulties identifying TMT labeled peptides when there is an extra N-terminus introduced. So it makes it easier to analyze the data, and that I think is also a big advantage.
Absolutely, thank you, Dr. Demmers. The next question would be for Alissa. Is there an HS version for the other PTMScan products like methylation, and how do they compare?
So at this time, we only have an HS product for KGG. We're actively working on developing HS products for all of our other products, or certainly starting from the most popular ones. So the next product that we'll be working on is phosphotyrosine, and I cannot give you an exact date of when that is going to be released, but that should be coming up next year.
Mm-hmm. A question for Jeroen. Why is there a difference in label-free quantification versus TMT?
You mean in terms of numbers of diglycide peptides?
Yeah, in regard to the number of diglycide peptides that are identified.
Yeah. So this is something that we actually only see when using the, let's say, the old version of the beads. When we use the new beads, we basically don't see any difference in numbers of diglycide peptides. Yeah. So I think that's positive.
Yeah, that's great. While we're speaking to you, another question for you, Jeroen. Can the HS antibody be reused?
Yeah, that is something that we haven't tried yet, but it's on the list. So that's something we are going to try soon, hopefully. Yeah.
All right. Alissa, urea is popular. Why is it so much underperforming compared to SDS?
That's a great question. I believe urea has been popular in proteomics just partly because detergents are very difficult, have historically been difficult to remove from samples, and they interfere with mass spec analysis. Biologists who aren't using mass spectrometry, have found benefits to all kinds of different detergents for specific applications, and SDS seems to just do a really excellent job of solubilizing cells and all of the various intracellular organelles.
Okay, thank you very much. Another question for you is about SUMOylation. So what is the return on search engine identification after a valve digestion? Augmentation, cleavage, higher charge on peptide, etc. usually decrease largely the rate of identification.
So there's a couple of aspects to that question. So when we do a whelp digestion, at least for us, we perform a search that's not enzyme specific. I think our search engine just doesn't have a whelp specificity programmed in. So, I mean, we're able to, we still can apply a 1% false discovery rate and identify, hundreds to a few thousand SUMO sites typically after a whelp digestion. SUMO whelp has more missed cleavages than does trypsin, but it cleaves at threonine, serine, alanine, and valine, which combined are very abundant amino acids. So if you had a situation where you could digest completely at every single one of those sites, you would end up with very small bits of peptide that might be too small for LC-MS analysis in many cases. So a few missed cleavages don't hurt you in that case. That question also mentioned higher charge on peptides. So there is going to be an increase of one charge state, but it's not. It shouldn't be that substantial. I mean, it works okay for us. So you are going to have higher charge states, but it should still work with a modern Orbitrap instrument.
All right, great. There are a few more questions, but keeping time in mind, I'll have to unfortunately stop here. If we haven't, we haven't been able to answer all the questions today, but we will get back to you by email as soon as possible on those. I'd like to thank the speakers once again for taking the time, and I also thank everybody for taking your time to join us today. I really hope that you have now a good understanding of our new PTMScan HS workflow and how you can use it to take your research to the next level. Well, have a good rest of the day or a good night, depending on where you're living. Thank you. Bye-bye.
Bye.